which is about investigating the relationship between the gene variant BH-LHE41 and the early onset of Alzheimer's disease. My name is Victoria Stanisławska.
I'm Samantha Rowan.
I'm Ravina Dipar.
I'm Mary Kubrick.
I'm Louisa Nacho.
I'm Hannah Lowe.
I'm Chloe Ritchie.
And I'm Anita Prussia. So here's a brief overview of our table of contents. This is what we're going to cover for today. So we have our intro, our background, our significance, methodology, limitations and implications, as well as ethics.
So our question that we're going to be examining today is how does the expression of the genetic variant BH-LHE41 correlate to the development of Alzheimer's disease? Background information.
This study aims to find a correlation between the gene BH-LHE41 and the risk of developing Alzheimer's disease.
Sleep is an essential part of our health and wellness. And as we know, most people need an average of 7 hours of sleep each night for optimal behavioral and cognitive performance. And sleep has many benefits, including memory recall, regulation of metabolism, and reducing mental fatigue. And even though there are many benefits, there are also obviously many consequences if we do not get enough sleep. And consequences associated with chronic sleep loss can be extremely dangerous and includes not only cognition impairments, but also emotion and physical impairments as well. Recent studies have shown that the effects of sleep restriction have the same effects of total sleep deprivation. And when sleep is disrupted, the active metabolic processes of the glymphatic system do not have time to accomplish their role. This results in the accumulation of toxic materials, and the repercussions express themselves through impairments in cognition, judgment, and conscience.
BH-LHE41 is a basic helix nucleus, family member E41. It belongs to a family of VHL-H transcription factors and DEC2 related to the Drosophila hairy cancer of split proteins. It has 8 genes spread over 50 kilobases on the Drosophila third chromosome. The hallmark of these gene complexes is that within any complex, the genes are evolutionarily related and jointly regulated. BH-LHE41 is linked to and believed to be involved in the circadian rhythm and cell differentiation. Proteins compete for specific E-box binding sites in the promoter of PER1. E-boxes appear in a broad variety of promoters and enhancers and are served as protein binding sites. They act as activators of transcription as well as repressors. They repress the clock/ARNTL's transactivation of PER1. And defects in this gene are associated with a short sleep phenotype, broadly found and expressed in thyroid and brain tissue